Hello everyone, welcome to Microbiology quiz. Topic is sterilization. After each question, there will be a pause for 5 seconds for you to think the answer and evaluate yourself. There is an explanation for each answer. You can pause the video to read the same. Let's start the quiz. Question number 1. The most appropriate method to sterilize a batch of culture media in sealed glass flask is A. Dry heat in a hot air oven at 80 degrees Celsius for 1 hour. B. Autoclaving at 121 degrees Celsius and 15 psi for 15 to 20 minutes. C. Filtration through a 0.45 micrometer membrane filter. D. Boiling at atmospheric pressure for 30 minutes. And the answer is B. Question number 2. A laboratory needs to sterilize a heat-sensitive vitamin solution. Which method is most appropriate? A. Autoclaving at 121 degrees Celsius for 15 minutes. B. Dry heat at 170 degrees Celsius for 2 hours. C. Boiling the solution for 30 minutes. D. Membrane filtration through a 0.22 micrometer filter. Answer is D. Question number 3. Which of the following best defines sterilization in microbiology? A. Reduction of microbial load to a safe level. B. Inhibition of microbial growth without killing cells. C. Elimination of all pathogenic microorganisms but not spores. D. Complete destruction or removal of all forms of microbial life including endospores. And the answer is D. Question number 4. Which biological indicator is commonly used to validate most heat sterilization in an autoclave? A. Bacteriophage suspension on agar. B. Spores of Bacillus subtilis on strips. C. Live culture of E. coli in broth. D. Spores of Geobacillus styrothermophilus in ampules or strips. Answer is D. Question number 5. Which type of material is best suited for sterilization by dry heat in a hot air oven? A. Aqueous culture media. B. Plastic petri dishes. C. Glass pipettes and metal instruments. D. Heat sensitive antibiotic solutions. And the answer is C. Question number 6. Which statement best distinguishes disinfection from sterilization? Both are interchangeable terms in clinical microbiology. Disinfection reduces or eliminates many pathogenic microorganisms but does not guarantee removal of all spores, whereas sterilization removes all viable forms. Disinfection kills only viruses while sterilization kills only bacteria. Disinfection eliminates all forms of microbial life, including spores, whereas sterilization reduces them to safe levels. Answer is B. Question number 7. Which property of ethylene oxide gas makes it useful for sterilizing certain medical devices? A. It sterilizes by simple dehydration only. B. It acts rapidly at very high temperatures. C. It is a non-toxic gas to humans at all concentrations. D. It can penetrate packaging and complex devices. at relatively low temperatures? And the answer is D. Question number 8. Which of the following is a major disadvantage of using ethylene oxide for sterilization? A. Ineffectiveness against bacterial spores. B. Requirement for very high sterilization temperatures. C. Requirement for long aeration time due to a toxic residues. D. Inability to penetrate packaging. Answer is C. Question number 9. Why is an exposure to ultraviolet radiation at 254 nanometer considered unreliable as a sole method for sterilizing surgical instruments? A. UV radiation has poor penetration and mainly affects exposed surfaces. B. UV radiation at this wavelength is completely harmless to microorganisms. C. It only kills bacterial spores and leaves vegetative cells intact. D. UV radiation instantly vaporizes any organic material present. And the answer is A. Question number 10. Which of the following best explains why prions present a challenge for conventional sterilization methods? A. Prions are living bacteria with thick cell walls. B. Prions lack nucleic acid, relying on abnormal proteins that resist standard inactivation. C. Prions are easily killed by low-level disinfectants. D. Prions are viruses with lipid envelopes that autoclaving cannot disrupt. Answer is B. Question number 11. Which sterilization method is most appropriate for sterilizing pre-packaged heat labile enzyme test kits used in diagnostic microbiology? A. Standard steam autoclaving at 121 degrees Celsius, simple refrigeration at 4 degrees Celsius, dry heat in a hot air oven at 170 degrees Celsius, and D. Gamma radiation or other suitable ionizing radiation. Answer is D. For watching the video. If you like it, don't forget to like, share and subscribe.